we are going to discuss about the unit of kidney which is nephron. So remember this, in our kidney we have 10 lakh nephrons are present in only one kidney. This means in the two kidneys we have basically 20 lakh nephrons. 20 lakh nephron. So now let's see the structure of nephron first. Yep, so this is basically our first portion which is glomerulus. Then this is Bowman's capsule. After this Bowman's capsule, this will lead towards a folded structure known as proximal convoluted tubules, or we can say PCT. Now then after that this PCT extend towards down, and again it will go towards up. This is basically loop of Henle. I'm gonna label it after this. After that, here we have another convoluted structure that is known as DCT, and now this DCT basically open into the duct which is known as collecting duct. Collecting duct. Okay, so if I can label this, so here this is basically yes, this is glomerulus. This is Bowman's capsule. Bowman's capsule. This is proximal convoluted tubule CT. This is loop of Henle, and loop of Henle basically contain two parts. This is descending loop of Henle, and this is ascending loop of Henle. Ascending loop of Henle. After that, this is basically an again convoluted structure known as DCT. It opens into collecting duct. So this is basically the structure of nephron. So now here you can see, yes. After in the glomerular structure, basically this is the afferent arteriole through which the blood enters. So I can write here afferent arteriole. Afferent arteriole, and this blood basically leaves through this artery which is known as efferent arteriole. Efferent arteriole. So now here we can say we know that artery or vein do not basically perform the function of diffusion. डिफ्यूजन का फंक्शन हमारे पास आर्टरीज और वेन्स के अंदर परफॉर्म नहीं होता है. तो बेसिकली हियर आर द कैपिलरीज इन द ग्लोमेरुलस व्हिच परफॉर्म द फंक्शन ऑफ diffusion. So these capillaries are known as glomerulus, and this here at this time basically the ultrafiltration will take place. We're gonna discuss these in detail, but let me mark here ultrafiltration. Okay, so yes, and so this is all. Now we can say right here the main points, what those all are. So yes, first we know that, okay, one more thing I forgot to mention over here. Yes. After this, here you can see through this efferent arteriole, this will basically further this will cover this whole process. ये इस process पूरे को cover करती है, and this will move towards the down. ये down तक भी move करती है. ये हमारे पास vasa recta को भी cover करती है. So the basically we can say these are paratibular capillaries which cover all the nephron. Paratibular paratibular capillaries. Now these paratibular capillaries when goes to the loop of Henle, so those are known as vasa recta. Vasa recta. Okay, so now here we can say let's discuss some points. Yes, the first point is all capillaries basically combine and they form the vein. We know all capillaries combine and they form the vein. Then another point is we know that the glomerulus and basically the Bowman's capsule. Bowman's capsule, as you can see, and this is glomerulus and this is Bowman's capsule. They both combine together and they form the structure which is known as Malpighian tubes, Malpighian body, or we can say tubes, Malpighian body. Now one more here structure, one more here basically topic is remaining. Now we are going to discuss about types of nephrons. Types of nephrons. So we have two type of nephrons. Yes. Okay, the first type of nephron is cortical nephron. Cortical nephron. Another nephron is just a medullary nephron. Just a medullary nephron. So if I can mark here the percentage of these both nephrons, so that we can say the percentage of this cortical nephron in our body is basically 85%, and the percentage of just a medullary nephron is 15%. So as the word defines, this is a cortical nephron and this is the juxtamedullary nephron. So this cortical nephron, this means this is present where this is present in the cortex of kidney, as we have discussed the structure of kidney. So this is present in the cortex of kidney. And this basically juxtamedullary nephron basically help to control the concentration of urine. This help to control the concentration of urine. How? Like it have to control the concentration of urine. Either the urine is dilute or concentrated. Okay, now we can say let's see what's the difference between basically the cortical nephron and just a medullary nephron. So if I can make here the nephron, sorry, kidney. Here you can see this is the kidney, and we have discussed that kidney have two parts. This is another part. Second part is known as medulla. First part is known as cortex. Cortex. Okay. So now here the nephrons are present. Okay, the nephrons are present someone like this. This is glomerulus, Bowman's capsule, PCT, loop of Henle, DCT, and collecting duct. Okay, over here it is also present PCT, loop of Henle, DCT, collecting duct. And here we have also this is glomerulus, this is PCT, loop of Henle, DCT, and duct. So these are known as cortical nephrons, which are basically only present in the cortex of kidney. So if we can discuss about what are the juxtamedullary nephrons then. So here you can see, as we know here the pyramids are present. So juxtamedullary nephrons are basically those nephrons whose loop of Henle go down towards the medulla, towards the pyramid. ऐसे nephrons हैं जिसका loop of Henle पूरा medulla के अंदर जाता है, पूरा मतलब pyramids के अंदर जाता है. So those are known as juxtamedullary nephrons whose loop of Henle go to go to medulla. Those are known as juxtamedullary nephrons. Okay, so I hope this is clear now. If you can, yes, till now it is clear. In the next video, we'll discuss about the act of maturation, like how the urine is produced.